Hello class, in this lecture we're going to talk about the process of diffusion, and we will save osmosis for a later lecture video. So let's begin. So before we get into the actual process of diffusion, there's three terms that I want to go over and reinforce with you. We did discuss these earlier this year, and they are solute, solvent, and solution. Alright, but before we get to those three, first off, what does concentration mean? Because this is going to directly affect the rate of diffusion later on. All right, concentration refers to the amount of what we call a solute within a solution. And before I touch on that, let me just bring up the rest of these three terms here. So again, concentration means how much solute within a solution. So what's a solute? So a solute is going to be what you're trying to dissolve in a liquid for our purposes during our lecture on diffusion here. So over here you can see a bowl of salt. Alright, salt is a solid, so that is going to be our solute. We're going to add that to water and try to dissolve it or mix it in. Alright, the liquid, water in this instance, is going to be now what we call the solvent. Alright, so it doesn't have to be water, it could be other things like hydrochloric acid or dozens of other possible liquids. But in this instance, water is going to be the solvent, which is going to dissolve the solute, which is salt. Could be sugar also. When the two of these are mixed evenly together, like you see here in the third bottle now, we have what's called a solution, which is an even mixture of both the solvent and the solute. So again, three S words which are there just to exist to annoy us, I'm sure, but we need to know what the difference between those three are, because it's going to be very important, and when we really start talking about how the concentration of a solute can affect bodily function, we're going to learn that it is always critical to maintain a correct balance of solute to solvent, always in our body. And the key example we've been discussing since the beginning of the year is sugar or insulin levels within our blood. We never want to have too much sugar or too little sugar. We never want to have too much insulin or too little insulin, because that could throw our body out of homeostasis. Again, before I move on, if I just address this picture here. Alright, so these two people here are going for a swim. If you've ever gone swimming for an extended period of time, been in the ocean for a long time, took an hour-long shower or bath, you've noticed that your fingertips start to prune, as well as the bottom of your feet can prune as well. The reason for that is those are the thinnest skin areas of your body, and any materials that are in your skin, alright, such as sugars, salts, etc., that are in higher concentration than they are in the water, they will diffuse out of your fingertips or toes, your feet, because the skin's thinnest there, out into the surrounding environment to try to reach a balance. So what you see is your fingertips start to shrivel because you are losing materials. Alright, so again, that's always going to be the case, that you're trying to reach a balance of solute in a solvent. Alright, so what is diffusion? You've heard this a million times. For our purposes, since we're focusing here on a cell membrane, it's going to be when particles move across a cell membrane, in particular from an area of high concentration to an area of low concentration. Alright, so if you look at this GIF up here, go back real fast. Alright, once it resets, you're going to have a high concentration of red particles right there, and now they are naturally diffusing from an area of high concentration to an area of low concentration. This happens naturally in any environment, liquid, gas, even solid. Solute, as long as they can, are going to want to move from high to low concentration. The same thing is going to happen, as you can see down here, across what we call a selectively permeable membrane. Selectively being that this plasma membrane here, or phospholipid bilayer, won't just let anything move from high to low concentration. It will be selective, but still we're going to be looking at movement from high to low. Now, before I get into the types of diffusion, I want to cover things that can actually affect the rate. When I say affect the rate, I mean control the speed. All right, is this particular factor going to speed up the rate of diffusion to reach balance, or is it going to slow it down, or is it going to stop it altogether? So the first one would be the size of the concentration gradient itself. When I say size of concentration gradient, I mean, alright, is there 90% sugar on one side of the membrane and only 10% on the other? Well, that's a high concentration gradient. So you're going to have a very quick movement of sugar from the 90% range over to the 10% range to reach a balance. But if you only had 60% sugar on one side of a membrane and 40% on the other, that's a very low concentration gradient. It's actually going to take, believe it or not, longer to reach balance on both sides of the membrane when you have a lower concentration gradient. The amount of cell membrane surface area. So the larger a cell doesn't really mean that it's going to be good for diffusion. In fact, once something moves into the membrane, it's going to take a longer period of time for whatever that particle or solute is to get where it needs to go inside a cell. So being a large cell is actually a negative compared to if you take up the same volume of space, but with many numerous cells. This is more efficient. This is why we're multicellular. So now anything that moves into the cell membrane doesn't have that much of a distance to go to get to where it needs to be. Versus here, where it could take much longer, the cell could actually die simply as a result of it waiting for food particles to get where they need to go, or waiting for waste to be expelled from the cell. What medium are we looking at for diffusion? Is it liquid, gas, or solid? Temperature, all right. As you heat up things, molecules begin to move faster, so the higher a temperature, the faster the rate of diffusion. The cooler the temperature, the slower the rate of diffusion. And then finally, what is the size as well as charge of a solute that's trying to get through the lipid bilayer? All right, both of those things could actually slow down the process. Again, just looking at another graphic, temperature, the higher a temperature, the faster the rate of diffusion. Surface area, the larger surface, you may have diffusion occur faster across the membrane, but as those particles are trying to move within the cell, actually the overall rate of diffusion is getting slower. So it's not a good thing to be a large cell. Again, higher gradient, faster rate of diffusion, smaller particles, faster rate of diffusion. And again, what is the medium? Solid obviously would be the slowest. It takes a long time for, say, water to percolate through rock. Liquid would be very fast. Gas would be the fastest. All right, but one thing I want you to take away is, once a concentration gradient no longer exists, that means you're not 90% on one side and 10% on the other. Now you're 50-50. You've reached what's called equilibrium. All right, and equilibrium refers to the process where you have equal amounts now of solute on both sides of a membrane. So there is no longer a concentration gradient. That does not mean diffusion stops. It simply means that you no longer have a mass movement from one side of the membrane to the other. You'll now just have an even exchange, 1 for 1. Again, another graphic here that you can see. Red particles high on the left, non-existent on the right. You're going to have a quick rate of diffusion until you reach equilibrium. While if you have now 5 red solute particles on one side and only 3 on the other, that's a lower concentration gradient. It'll actually take longer to reach equilibrium in this condition than it would have up here. And again, once you go from high to low, you reach equilibrium on both sides of the membrane. I have the arrow now facing both directions. Diffusion doesn't stop. You just no longer have a concentration gradient like you do in each one of these scenarios. All right, it's higher up here than it is down here. Alright, so now we can talk about the 2 types of diffusion. In this lecture, I'm only going to focus on the first of the 2. But the 2 types of diffusion that we're going to cover are called passive transport and active transport. Alright, in this lecture, we're just going to finish up now with passive transport. So if you hear, I'll become an English teacher for you. If you hear that, what does the term passive mean, class? Alright, I couldn't be an English teacher, but passive means very low energy, right? Not motivated, can barely do anything. Well, in this instance, passive is going to be a good thing. All right, passive transport is going to refer to when solute can move across the membrane from high to low concentration, but it doesn't cost a single cent. It's free. So you don't have to expend any of the ATP energy we're going to be talking about a lot more as we move into our next unit on photosynthesis and cell respiration. So passive transport is going to be free, no energy required, moving from high to low concentration. The only difference is going to be is this. How are these solute particles going to move from high to low across the membrane? Are they going to go directly through the phospholipid bilayer themselves, like you can see here where my laser pointer is? So we have a high concentration out here and a lower one in here. These solute are just moving directly through the phospholipids. Or can the solute not squeeze through the phospholipids for whatever reason? So now they have to make use of one of these integral membrane proteins to go from high to low. And again, I don't care what the scenario is. Are particles moving into the cell or out of the cell? It doesn't matter, as long as it's moving from high to low and it doesn't cost any energy. So we're going to give a name to these two mechanisms of passive transport. And that's our next slide here. So is it going to be what we call simple passive transport? Or is it going to be what we call facilitated passive transport? Simple is simple. It's going to be when the solute moves directly through the phospholipid bilayer without the use of energy. So they can squeeze their way through, whatever these solutes are, directly through the phospholipids. Generally, these are going to be very small molecules, only a couple atoms, but in particular, they will be nonpolar. Remember, nonpolar is when electrons are shared equally, no charges whatsoever. So since the hydrophobic tails in the middle of the membrane are also nonpolar, these molecules will not be repelled. Most times, these are types of gases like oxygen and carbon dioxide, which are both nonpolar and very small. So they can both move in and out directly through the lipid bilayer, unaffected, never slowed down. They're going to be able to move right through with no hindrance. While facilitated means that now you need some help. Facilitate means to help. So larger, or in this instance, polar molecules are going to have to go through the lipid bilayer, but since they can't squeeze through the lipid bilayer, they're going to have to use some of those integral membrane proteins that we discussed earlier. So larger molecules that are too big to fit through the lipid bilayer, like glucose, or ones, even if they're very small, but they have a charge, like a hydrogen ion, they're going to need to move through one of those integral membrane proteins in order to get from one side of the membrane to the other. Alright, so once again, simple is easy. High to low, right through the lipid bilayer, not costing any energy. And they're most times going to be gases, which means they're very small and nonpolar. While with facilitated diffusion, it's still high to low, still doesn't cost any energy. But now we have larger molecules or ones that have a charge that can't fit directly through the lipid bilayer, so they're going to need some help. All right, so we're going to use integral membrane proteins, which are going to act like tunnels through a mountain. So finally, let's visualize this. So if I just focus here on this diagram first, all right, we have simple on the left, and then we have two forms of facilitated here on the right. But again, both are forms of passive transport. Simple, you can see how they can go directly through the lipid bilayer, so they're going to be very small. And these molecules will also be nonpolar, just like the hydrophobic tails are. That'll occur until you reach equilibrium. Now we have particles or solute that maybe they're large, like glucose, or maybe they just have a charge, like a sodium ion, a chlorine ion, a hydrogen ion. They will be repelled by this hydrophobic layer, or they can't squeeze through the phospholipids themselves. So now we make use of these integral membrane proteins. All right, there's actually two types. There's open channels as well as what we call carrier proteins. I'm not going to distinguish between the two. They're just both going to aid moving solute high to low, either into a cell or out, through an integral membrane protein. And again, you don't see any ATP being used, so this is a form of passive transport. And again, if you just look over here, you can see some GIFs. Again, here is high to low until you reach equilibrium, simple passive transport. Or again, high to low until we reach equilibrium on both sides, but these particles would be too large or have a charge. They have to use the integral membrane proteins. All right, if you have any questions, make sure you ask me during our class period. Thank you.